Welcome to the world of medicine making. Have you ever wondered how the medicines we take are made? It's kind of like a super precise science experiment. To make sure every pill or liquid medicine is safe and works the way it should, scientists and doctors from all over the world came together to create a set of rules. This team is called the International Council for Harmonization, or ICH for short. Their job is to make sure that a medicine made in one country meets the same high standards as a medicine made in another country. Now let's zoom in on one of their most important rulebooks, called ICH Q14. The Q stands for quality, because that's what it's all about. ICH Q14 is a guide for developing analytical procedures, special tests that scientists use to check medicine, like being a detective for molecules. ICH Q14 gives scientists a modern, flexible, and scientific way to create these tests. It encourages them to really understand the science behind their tests, not just follow a recipe blindly. This is called a life cycle approach, thinking about the test's entire journey, from creation to daily use, and even to eventual retirement when a better test comes along. Why is this such a big deal? The tests that ICH Q14 helps create are the final gatekeepers that stand between a batch of medicine and the people who need it. If these tests are not accurate, a medicine that is too strong, too weak, or contains harmful substances could accidentally be approved, putting patients at risk. ICH Q14 ensures these tests are built on a solid foundation of science. When you see a medicine on the shelf, you know it has passed incredibly precise and well-designed tests. Having a global standard makes the entire drug approval process smoother and faster, creating a clear path for getting important drugs from the lab to people who need them most. Every great project starts with a great plan. The first step in the ICH Q14 journey is creating the Analytical Target Profile, or ATP, a clear description of what your test needs to do. For example, this test must accurately measure the active ingredient in our headache pill and tell the difference between the good ingredient and any leftover manufacturing materials. Creating the ATP is a team effort. Scientists, quality experts, and regulatory specialists ask important questions. What are we measuring? How accurate must it be? Are we looking for something big or tiny? This blueprint is a living guide that the team refers back to throughout development. It's their North Star, ensuring every analytical procedure is built with clear purpose and solid scientific foundation. After you have your blueprint, it's time to choose your tools. Think of a laboratory as a giant toolbox filled with amazing scientific instruments, each designed for a specific job. For measuring the main ingredient in a tablet, a common tool is high-performance liquid chromatography, or HPLC, like a super-powered filter that separates ingredients so they can be measured accurately. Choosing the right technique is critical. The team considers sensitivity, specificity, and practical factors like test duration and complexity, finding the perfect balance between scientific power and everyday usability. Once chosen, real experimentation begins. Scientists develop the specific recipe, running many small experiments and tweaking settings until the method works perfectly and reliably. Now it's time to think like a detective and look for potential problems. This crucial step is called risk assessment, asking, what could possibly go wrong with our test? The team brainstorms everything that could vary: temperature changes, chemical mixing variations, timing differences. These small variations are potential risks because they could affect test accuracy. After identifying key risks, scientists put controls in place. They perform experiments to understand high-risk factors better, like testing at different temperatures to define safe operating ranges. By understanding and controlling these risks, they create a tough, reliable test that gives the right answer every time, even with small, unavoidable variations of a busy lab. This is the moment of truth. Validation is the official process of proving, with hard evidence, that the analytical test does exactly what you designed it to do, like the final exam for the test method itself. Scientists demonstrate accuracy using highly pure reference samples with known amounts, checking if their measurement matches the true value, like checking a kitchen scale with a certified weight. They prove precision by running the same test repeatedly, showing consistent results every time, like a professional archer hitting the bullseye with every arrow. All validation data is collected in a formal report, official proof that the method works. This document is reviewed by quality experts and health authorities, confirming the procedure is ready for its important duty. Let's see ICH Q14 in action. Health First Pharma is developing vitality drops for elderly patients. They need to test for the active ingredient Energene in every bottle. First, they create their ATP, accurately measure Energene in a 95 to 105% range, and not be fooled by the sugary syrup used for taste. This clear goal guides their entire project. They select HPLC because it separates Energene from sugary syrup. During risk assessment, they identify pH as critical. Small changes could affect the Energene molecule shape and test results. They discover that keeping pH between 4.0 and 4.5 gives perfect, reliable results. This defined range is the method-operable design region, a key ICH Q14 concept that makes their method robust. How can you use ICH Q14 wisdom daily? Always start with the why. Write down your analytical target profile. Having a written blueprint that everyone agrees on saves headaches and keeps projects focused. Be a proactive problem solver. Don't wait for things to break. Embrace risk assessment. Brainstorm everything that could affect your test and ask what-if questions constantly. Document your journey thoroughly. Write not just what you did, but why you did it. This provides scientific justification and helps regulators understand your work. Remember, method development is a life cycle. Continue monitoring performance, investigate trends, and embrace continuous improvement. Following ICH Q14 principles creates a culture of scientific excellence, ensuring the medicines we depend on are the safest and highest quality possible.